So now that we've covered the basic location and structure of the gastrointestinal tract and the accessory digestive organs, we are now going to move on to how these structures all work together to take the food that we ingest, break it down into molecules that the cells of our body can absorb, and then expel the waste. There are only two learning objectives for the physiology of the gastrointestinal system. So firstly, to describe the processes of digestion that occur all the way along the gastrointestinal tract from mouth to anus, as well as the role of those accessory digestive organs. And then secondly, to describe the major hormones that regulate digestion, so bringing in a little bit of the content from the last series of lectures. Getting started with learning objective number 1, to describe the processes of digestion that occur from the mouth to the anus. So when I say the processes of digestion in this learning objective, I am referring to those 6 basic digestive processes that we spoke about in the previous lecture. So we had ingestion, secretion, mixing and propulsion, digestion, absorption, and defecation. These processes occur all the way along the gastrointestinal tract, from the mouth, then the pharynx, the esophagus, the stomach, the small intestine, and then the large intestine. And then along the way, those accessory digestive organs also play a part. So we have the teeth, the tongue, and the salivary glands, which play a role in the mouth. We then have the liver, the pancreas, and the gallbladder, which play a role in the small intestine. So if we start at the mouth, and remembering that the mouth includes not only the oral cavity, but also the teeth, the tongue, and the salivary glands. And the digestive processes that occur in the mouth include ingestion. So we take our food and drink and we put it in our mouth. We have mechanical digestion, which results from chewing or mastication. This is where the food is manipulated by the tongue, it's ground up by the teeth, and it's mixed in together with the saliva. As a result of this chewing, the food is reduced to a soft, flexible, easily swallowed mass called the bolus. We then have chemical digestion. So this is where enzymes that are secreted into the saliva are mixed with the food and start to break it down. So in the mouth, we have 2 enzymes that are released. We have salivary amylase, which initiates or it begins the breakdown of starch, which is a type of carbohydrate. We also have lingual lipase, but this needs to be activated by the acid of the stomach before it actually begins to be effective, so we don't talk about this one quite as much in the mouth. Thinking a little bit about our saliva, so our saliva is mostly made up of water. It's 99.5% water, with that remaining 0.5% made up of various ions, some of which can buffer any acidic food that enters the mouth. It's also made up of some dissolved gases, and then of course the enzymes that I just mentioned. The saliva is not only important for those enzymes to begin chemical digestion, but also to lubricate the food and make it easier to move around the mouth and then swallow. Now, the autonomic nervous system controls the production of saliva, so it's not something that we have to think about, but it can be stimulated by a range of things, including the smell, sight, sound, or even the thought of food. Lastly, for the digestive processes of the mouth, we have propulsion, and this is purely related to swallowing. So the tongue forces the food bolus to the upper back part of the mouth, ready to be swallowed into the oropharynx. So the digestive process that occurs in the pharynx is quite straightforward. It's all related to swallowing. So food passes from the mouth into the oropharynx, then the laryngopharynx, and it's the skeletal muscle contractions of these areas that propel the food down towards the esophagus and then into the stomach. Similarly, the single digestive process that occurs in the esophagus is propulsion, and the esophagus moves the food from that laryngopharynx to the stomach via swallowing, which you may also see referred to as deglutition. So because it is important to help us ingest our food, we're going to take a little bit of a closer look at swallowing. And this process is facilitated by the secretion of that saliva and the mucus in the mouth. And it involves the mouth, the pharynx, and the esophagus all together. So swallowing actually occurs in 3 stages. We have the voluntary phase, which you might also read as the oral stage. And this is when the bolus of food is passed from the mouth into the oropharynx. We have the pharyngeal stage, which involves the involuntary movement of the bolus through the pharynx into the esophagus. And then the esophageal phase, which is where that bolus of food moves from the esophagus into the stomach. So if we look at that first stage, swallowing starts when that bolus of food is forced to the back of the mouth and into the oropharynx by the movement of the tongue. The tongue goes upward and backward against the hard palate, and that food is forced into the oropharynx. This stage of swallowing is considered voluntary because the movements are controlled by skeletal muscle. So our tongue is skeletal muscle, so it's under voluntary control. The pharyngeal phase begins when the bolus moves into the oropharynx. So remember, the oropharynx is this part of the pharynx here, which sits behind the oral cavity or the mouth. And when that bolus moves into the oropharynx, it actually stimulates receptors, which then send an impulse to the deglutition or the swallowing center in the brainstem. The neural impulse, which returns from the brainstem, then causes the soft palate and the uvula to move upward and close off the nasopharynx. So remember, this is your soft palate here. That very end is the uvula. That impulse from the brainstem causes it to move upward, so that food can only go down in this direction and not up into that nasal cavity. At the same time, our larynx, which was this flap of cartilage here, will close off the trachea. So again, the food can only go in one direction, down into the esophagus and not down into your lungs. Now, this pharyngeal stage is involuntary because these actions occur as part of a reflex. So a reflex is when the neural impulse goes to the spinal cord or the brainstem and not the brain itself, and it's not something that's under conscious control. As the bolus moves through the oropharynx and then the laryngopharynx, our upper esophageal sphincter will relax and the bolus will then enter the esophagus. So that's when we have our esophageal phase. During this stage, peristalsis, which is a particular contraction that we'll talk about in a second, pushes the food down into the stomach. This contraction occurs with smooth muscle, so it's again not under voluntary control. So we've mentioned peristalsis already today, and I think we talked about it a little bit in the previous lecture, but peristalsis is a particular type of contraction that we see all the way along the gastrointestinal tract. It's an automatic wave-like movement of smooth muscle. And in this type of contraction, the inner layer of circular muscle behind the food will contract, while the inner circular layer of muscle in front of the food will relax. At the same time, we've got longitudinal muscle that runs down here, and as the food enters that region, it will also contract to help move this food in one direction. Now, I apologise if anyone is a bit squeamish, but I did want to give you a bit of a visual as to what peristalsis actually looks like. So this is peristalsis occurring in the small intestines. And so you can see that it's that wave-like contraction of the smooth muscle, and it's pushing food down the gastrointestinal tract in one direction. Kind of looks like how a snake might swallow its food. But moving on to the stomach, and the digestive processes that occur in the stomach are mechanical digestion, secretion, and chemical digestion. And we're going to talk about each of these in a bit more detail. So starting with mechanical digestion, And a special feature of the stomach is that deep to the mucosa and the submucosa, the muscularis layer of the stomach actually has 3 layers or 3 sheets of muscle, instead of the 2 that we typically see elsewhere throughout the gastrointestinal tract. So we've got our outer longitudinal layer that we see elsewhere in the gastrointestinal tract. We have the inner circular layer, but what's unique to the stomach is this very inner oblique layer of the stomach. So several minutes after food enters the stomach, we have waves of peristalsis about every 15 to 25 seconds, and each wave of peristalsis moves the contents of the stomach down towards the pyloric sphincter. So if we take a look at this stomach here, it will enter through the esophagus. That peristalsis will now push the contents of the stomach down towards the pyloric sphincter before it enters the duodenum or the small intestine. The movement of the food down towards the pyloric sphincter is known as propulsion. Now, because most of the food particles are initially too large to fit through this narrow sphincter, they're then forced back up into the stomach in a process called retropulsion. So it's come down along here with those waves of peristalsis. It's too big to fit through the sphincter, so it's hit the end and then come back up into the stomach. That process is retropulsion. Now, this process will continue to happen and happen until that food is now like a nice liquidy soup. Those food particles are actually small enough to exit through the pyloric sphincter. As food exits the pyloric sphincter into the duodenum, we call this process gastric emptying. So taking a closer look at some of those terms, because they are important terms to be familiar with, we have propulsion, which is just the movement of food from the esophagus down towards the pyloric sphincter. It occurs because of those waves of peristalsis. We then have retropulsion, which is where those food particles are too big to pass through the sphincter. So it hits the sphincter and comes back up into the stomach, and that's retropulsion. We then have gastric emptying, which is the process by which chyme, so once the contents of the stomach are in that nice liquidy soup-like consistency, we now call it chyme. You can see that spelled here. Once that food is small enough to exit the pyloric sphincter into the duodenum, we now call that process gastric emptying. And gastric emptying is a really slow process, with only about 3 milliliters of chyme moving into the duodenum at any one time. So a little bit more on gastric emptying, because this is going to form the basis of the experiment that we complete in class for this lab. So as I said, gastric emptying is the process by which chyme moves from the duodenum through the pyloric sphincter into the duodenum. We need to regulate this process because it takes a lot longer to digest and absorb our food than it does to ingest it. So we can eat really quickly and the food can sit in our stomach, but to actually break down that food so that we can absorb the nutrients takes a much longer period of time. And now there are a few factors that can either increase or decrease the rate of gastric emptying. So looking at things that increase gastric emptying, or how fast the contents of the stomach will leave the stomach and go into the duodenum, and we have things like the distension of the stomach. So that's just how full you are. So remember, when you eat, your stomach will fill up. Those gastric rugae will unfold and your stomach expands. How full or expanded your stomach is will influence the rate of gastric emptying. The more distended, it means that we have more food ready to be digested and then absorbed. So we want to get this process moving so it will increase gastric emptying. The presence of liquid in the stomach will also increase the rate of gastric emptying, as this means that the ingested food is already in the liquid chyme form that it needs to be in to pass through that narrow pyloric sphincter. So we don't need to do any of that mechanical digestion to kind of break it down into this form. Factors which will then delay or decrease gastric emptying are the presence of solids in the stomach. So this means that the food has not been mixed and churned. We haven't had enough propulsion and retropulsion to break down the food so it's in a consistency that can actually pass through that narrow pyloric sphincter. Distension in the duodenum. So if there's already a lot of chyme in the small intestine that still needs to be digested and then absorbed, the body will try and hold onto the food that it has in the stomach so that we don't overload the small intestine. And then the presence of fat or protein in the duodenum. So fat in particular is a little more complex to digest and absorb than, say, a carbohydrate. And so when we have a meal that's high in fat or high in protein, our body will actually delay how quickly this moves into the small intestine so that it allows our body time to produce the additional secretions that our body needs to actually digest and then absorb these substances. And so we'll talk more about this in particular when we come to the function of the liver, the gallbladder, and the pancreas. Coming back to this slide though, and our next digestive process that occurs in the stomach is secretion. And we have several cells within the stomach that facilitate this function. So as we've already discussed, the stomach is made up of the same basic layers as the rest of the gastrointestinal tract, but with a few modifications. We've already talked about the muscularis layer, so that we have those 3 layers of muscle instead of 2. But the innermost layer of the wall of the stomach is also structured a little differently. So the mucosa of the stomach is a little different. So the innermost layer of the mucosa is made up of simple columnar epithelial cells. So one layer of column-shaped cells called surface mucous cells. These surface mucous cells will extend down into the lamina propria, where they form columns of secretory cells called gastric glands. Several gastric glands will open up into a narrow channel, which we now call a gastric pit. The secretions of several gastric glands flow into each gastric pit. Geez, that's a bit of a tongue tie. And then into the lumen of the stomach. So we have these gastric glands. They each have various secretory cells in the walls. They then come into this gastric pit, which then opens up into the stomach to help mix with all of our food. So our gastric glands contain 3 types of exocrine glands that secrete their products into the stomach. We have mucous neck cells, we have parietal cells, and then we have chief cells. Our gastric glands also contain one type of enteroendocrine cell, which is just an endocrine cell of the stomach. This is called our G cell. So both our surface mucous cell and our mucous neck cell, which as the name suggests, secrete mucus. The main function of secreting mucus is to protect the underlying tissue of the stomach and then help combine the food all together and then move it throughout the rest of the gastrointestinal tract. Our parietal cells secrete hydrochloric acid and intrinsic factor. Intrinsic factor is needed for the absorption of B12. And then we have our chief cells, which secrete pepsinogen and gastric lipase. The secretions of these 1, 2, 3 cells here together is what we call gastric juice. And we produce 2 to 3 litres of gastric juice per day. The G cell secretes the hormone gastrin into the blood, and we'll actually look at that hormone in the second learning objective. Now, the last digestive process which takes place in the stomach is chemical digestion, and this comes about as a result of the secretions from the cells that we just mentioned. So, kind of thinking about that process as a whole, our food will enter the stomach from the esophagus, and aside from participating in the mechanical digestion, secretion, and the chemical digestion, one of the main functions of the stomach is to act as a temporary storage site. So the food can actually sit in your stomach for a number of hours before you actually start churning and mixing up the food. During this time, before you start that propulsion and retropropulsion, the carbohydrate within the food will start to become digested thanks to the salivary amylase that we secreted into the saliva and mixed with the food in the mouth. However, once we start mixing our food with our gastric juice, that salivary amylase actually becomes inactivated due to the acidic nature of the gastric juice. So the salivary amylase will only digest the carbohydrate before we start mixing the contents of the stomach with that gastric juice. However, our linguinal lipase, which is another enzyme that we release into the saliva, this one breaks down fat. It's inactive in the mouth and it's inactive when the food is just sitting in the stomach. But once it mixes with that acidic gastric juice, it then becomes activated and it can now start breaking down fat. We then have our parietal cells, and these cells secrete hydrochloric acid. This is what makes that gastric juice really acidic, and its main job is to kill many of the microbes in our food. Hydrochloric acid also partially denatures the protein that we ingest, as well as stimulates the secretion of a number of hormones which promote the release of bile and the release of pancreatic juice. The hydrochloric acid also activates pepsinogen, and so pepsinogen is secreted by our chief cells. When pepsinogen comes into contact with the hydrochloric acid, it's converted into pepsin, which is an enzyme that begins to break down protein. The chief cells also release gastric lipase, which breaks down fat, but in the stomach, this only has a very minor role. Now, not fully related to chemical digestion, but worth mentioning while we're talking about the stomach, only a very, very small amount of nutrients are actually absorbed in the stomach, mostly because we haven't done the majority of our digestion, so it's just not in the form that we can absorb into our cells. What is absorbed in the stomach is a very, very small amount of water. Certain drugs, like aspirin, will be absorbed in the stomach, and that's why aspirin and our Panadols can start working really quickly, and also alcohol. Again, why if you drink quite a lot on an empty stomach, you'll start to feel those effects of the alcohol really quickly. So continuing on down our gastrointestinal tract, and before we come to the small intestine, we're going to have a look at those accessory digestive organs that secrete into the small intestine and then help with the chemical digestion in the small intestine. So first up, we have the pancreas, and the digestive process that occurs in the pancreas is secretion. And so remembering from our last lot of lectures that the pancreas is both an endocrine gland that secretes hormones, as well as an exocrine gland that secretes digestive enzymes. So the pancreas is made up of clusters of epithelial cells. About 99% of those clusters are called acini. These make up the exocrine portion of the organ, so that's these yellow cells around here. The cells within the acini secrete a mixture of fluid and digestive enzymes that we call pancreatic juice. And we make about 1.2 to 1.5 litres of pancreatic juice per day. Our pancreatic juice consists mostly of water, of salts, of bicarbonate, and of enzymes. The bicarbonate functions to help buffer the acidic gastric juice that comes from the stomach. The enzymes is what helps break down our food. So of those enzymes, there's three that I want you to be aware of. We have our pancreatic amylase. So we have salivary amylase, which is released in the mouth. We have an amylase that is released by the pancreas. This helps break down our carbohydrates. We have trypsin, which digests proteins. And we have pancreatic lipase, which digests our fats. So remember, we also have gastric lipase, which is secreted by the stomach, but only plays a very minor role. Now, like pepsin, trypsin is actually secreted in an inactive form called trypsinogen, But when it comes into contact with an enzyme on the microvilli of the small intestine, so where it's secreted into, it's broken down into this active form called trypsin, so then it can become effective. Now, the remaining 1% of clusters of cells within the pancreas are called our pancreatic islets. These make up mostly our alpha and our beta cells. As we discussed briefly last week, our alpha cells secrete glucagon, which work to increase our blood glucose concentration, and our beta cells secrete insulin, which help reduce our blood glucose concentration. The other two accessory digestive organs that secrete their products into the small intestine are the liver and the gallbladder. The liver produces bile, and it can either secrete it directly into the small intestine, or the bile can be stored and concentrated in the gallbladder before it then is released into the duodenum. Aside from secreting bile, the liver also plays a role in carbohydrate metabolism, so it can store glucose as glycogen when we need to reduce our blood glucose concentration, or it can break down that glycogen and put it back into the blood as glucose when we need to increase our blood glucose concentration. It also can break down triglycerides or fat to produce ATP, as well as then store those fatty acids when required. The liver also detoxifies the blood from substances like alcohol, drugs, and hormones. It can break down old red blood cells, assist in the production of vitamin D, and store vitamins like vitamin A, B12, E, and K. The main function of the liver in relation to the gastrointestinal system, however, is that the liver produces bile. And so each day, the hepatocytes of the liver, which are the cells that the liver is made up of, secrete up to 1 litre of bile. Now, bile is a yellowy green liquid that consists of water, of bile salts, of cholesterol, of bile pigments, and of several ions. The principal bile pigment is bilirubin, which is produced by the phagocytosis of old red blood cells. This bilirubin is what gives bile its distinctive colour. So if you go into the labs and take a look at some of our cadavers, you'll be able to identify the gallbladder really easily because it is a really bright green colour thanks to that bile. You'll also know if you've been really sick and you've had nothing in your stomach but you continue to throw up, you start to throw up that kind of yellowy greeny contents. That is some of the bile coming out into your stomach. Now, the role of bile in the digestive process is to emulsify or break down large lipid or fat globules into small lipid or fat globules. These smaller globules then present a larger surface area that allow the pancreatic lipase to more effectively digest the lipid molecule. Without the presence of bile, very little fat digestion would take place. So looking at in a visual form, if this is a big droplet of fat, if our lipase was to try and attack this globule of fat, it would be very ineffective because there's a lot of fat in here and not much surface area for it to kind of eat away at. That's where bile comes into place. So we have bile, which is made up of bile salts and a number of other ingredients. It breaks our big fat droplet into smaller fat droplets and then smaller again. We now have a less fat in the middle to a greater surface area that the lipase can then attack the fat, which makes it much more effective in breaking the fat down. Arriving now at the small intestine and 4 digestive processes that occur in this region, and we have mechanical digestion, which takes place in the form of 2 specialized types of muscle contraction. We have chemical digestion, which occurs mostly due to the release of bile and pancreatic juice. We have secretion of various endocrine and exocrine substances from cells in the mucosa, And then we have absorption, remembering that most of the absorption of nutrients in our body occurs in the small intestine. So the small intestine plays a role in mechanical digestion due to two specific muscular contractions. These are called segmentation and then peristalsis, which we've already talked about. Segmentation is localized mixing contractions that occurs in portions of the small intestine that are distended by a large volume of chyme. So if our small intestine is really full, we have segmentation to kind of break it down into smaller parts. Segmentation mixes the food, but it does not move it along the gastrointestinal tract. So segmentation begins with the contraction of circular muscles in one section of the small intestine, an action that will constrict the intestines into segments. So we've got a constriction here and a constriction here. Next, the muscle fibers that sit kind of within those segments will then also contract, dividing each segment of the small intestine into a smaller segment again. Finally, those first fibers to contract will relax and those smaller segments will unite. This kind of makes the food or the chyme slosh back and forwards, and as a net result, it becomes all mixed up. Peristalsis, on the other hand, which we've already talked about, is the progressive or wave-like contraction of muscle. This is what pushes the food along the gastrointestinal tract down towards the large intestine. So segmentation is kind of squishing it together into little chunks, mixing those sections of the small intestine back and forward to mix up the food. Peristalsis is what moves it from one end of the gastrointestinal tract to the other. So when the duodenum receives the chyme from the stomach, it's now a watery soup of partially digested carbohydrates, fats, and proteins. To completely digest these nutrients, we must have chemical digestion. And chemical digestion in the small intestine is largely a result of our secreted bile and secreted pancreatic juice. Once the chyme arrives in the small intestine, the carbohydrate is broken down by our pancreatic amylase, as well as enzymes that are located on the microvilli of our mucosa. So remember those tiny little fingers that extend out from the absorptive cells in our mucosa. Carbohydrate is broken down to a monosaccharide, which is the form that it needs to be in to be absorbed by the body. So we break our carbohydrates down into monosaccharides. For our protein digestion, we have enzymes in pancreatic juice which include our trypsin, as well as enzymes on the brush border or the microvilli, which will break down those proteins into single amino acids. Amino acids are the form that we need the protein to be in to absorb the protein. For the digestions of lipids, remember we kind of have that two-step process. So we have the emulsification of the fat by bile, and then the digestion of fat by the pancreatic lipase. Like the stomach, the small intestine also has a number of specialized cells that help carry out digestion and absorption, and therefore carry out both secretion and absorptive digestive processes. We have absorptive cells, which their function is nice and simple. It absorbs our nutrients. These are the cells that sit on the very inner surface of the lumen of the small intestine. They have the little microvilli on top to increase the surface area, so there's more contact for the nutrients to be absorbed. We have goblet cells, which secrete mucus. This helps protect the lining of the small intestine because the chyme can often be quite acidic, particularly coming from the stomach where there's lots of hydrochloric acid. We have a number of enteroendocrine cells, which remember are our hormone-secreting cells within the gut. These secrete hormones including secretin and CCK, which we'll talk about in the next learning objective. And then we also have Paneth cells, which secrete lysozyme. and we believe these play a role in immunity. But just coming back to absorption, because as I mentioned, most of the absorption of our nutrients occurs in the small intestine, and because of this, its structure is specifically adapted for this function. So for example, the length alone of the small intestine provides a really large surface area for digestion and absorption, but then we also have other structures which further increase that surface area. So we have our circular folds, also sometimes called the plicae circularis. These are folds of the mucosa and the submucosa. Some extend the entire way around the small intestine, some just partially a way around. These folds enhance absorption by 1, increasing surface area, and 2, causing the chyme to spiral around the small intestine instead of just passing through in a straight line. Again, as we've already spoken about, we have our villi. Our villi are the finger-like projections coming out of the mucosa. They're up to 1 millimeter long, with approximately 20 to 40 villi per square millimeter. Nutrients absorbed by epithelial cells covering the villi then pass through the wall into a capillary to enter the bloodstream. The small intestine also has microvilli, which remember are the projections on the top of absorptive cells, or it's the projections that come off of the villi. So if this is a villi, these little projections are the microvilli. This significantly increases the surface area with an approximate 200 million microvilli per square millimeter of small intestine. Because to the naked eye they are so small to see, this structure of microvilli is sometimes referred to as the brush border. The brush border contains several digestive enzymes. Lastly, we have the large intestine, which is the location of 2 digestive processes, of absorption and of defecation. So compared to the small intestine, the mucosa of the large intestine does not have any structural adaptations to increase surface area. There's no circular folds or villi, although they do have microvilli on the surface of any absorptive cells. Consequently, not as much absorption occurs in the large intestine as the small intestine. Most of what is absorbed in the large intestine is water, although relatively this is also quite low, with about 90% of the water already having been absorbed in the small intestine. Despite there being a relatively low amount of water absorbed in the large intestine, it still does make a really important contribution to maintaining our body's water balance. And aside from water, the large intestine also absorbs really small amounts of ions and certain vitamins. Now, after the chyme sits in the stomach for a while, it goes through the small intestine. It becomes a solid or semi-solid state because all the nutrients and the water have been absorbed. Once it reaches the large intestine in that semi-solid state, we now call it feces. Feces is made up of water, of salts, of any unabsorbed material that was digested, or any material that we can't actually digest, for example, fiber our body can't digest, as well as epithelial cells, which are actually sloughed off as the chyme, or the now feces, moves through the mucosa of the gastrointestinal tract. In the mucosa of the large intestine are many goblet cells. Remember, our goblet cells secrete mucus, and they lubricate the pathway for feces. There is a very small amount of chemical digestion that occurs in the intestine, but I haven't listed it as a specific process because not a lot occurs. And it doesn't actually occur because of the large intestine itself, but rather the bacteria that lives in the large intestine. This bacteria will ferment any carbohydrate that is left within the feces, and as a result, it can release gas. and it is this gas that will contribute to flatulence. So taking a close look at the digestive process of defecation, and a particular contraction of the large intestine called mass peristalsis moves the feces from the sigmoid colon into the rectum. The resulting distension or stretching of the rectal walls will stimulate stretch receptors, and these initiate a defecation reflex. So a portion of defecating or going to do number 2 in the toilet actually happens automatically and without conscious involvement. So in response to the stretching of those rectal walls, the sensory nerve impulses are sent to the sacral spinal cord. Motor impulses then travel along our parasympathetic motor nerves to the descending colon, to the sigmoid colon, to the rectum, and to the anus. This causes contraction of longitudinal muscles that runs along all of those parts of the large intestine. That causes shortening of those areas and increases the pressure in the rectum. That increase in pressure causes the feces to move towards the anal sphincter. We then have relaxation of the internal anal sphincter through that parasympathetic stimulation. So remember, our internal anal sphincter is made up of smooth muscle, so it's not something that we can have voluntary control of. Our external anal sphincter is made up of skeletal muscle and is therefore voluntary controlled. And if it is not consciously relaxed, then defecation does not occur. If defecation doesn't occur, that feces backs up into the sigmoid colon until we have the next wave of mass peristalsis, stimulating those stretch receptors again and then reinitiating that reflex. So for defecation to occur, we need to have that parasympathetic relaxation of the internal anal sphincter, as well as that conscious or that voluntary relaxation of the external sphincter to actually have that feces exit the body. Now, in infants and babies, the defecation reflex will cause automatic emptying of the anus because voluntary control of that internal anal sphincter has not yet been developed. When we have conditions like diarrhea, these sphincters are not actually affected, but it's an increase in the frequency, the volume, and the fluid content of the feces, because we've had the food or the chyme move too quickly throughout the intestines, and we don't have time to actually digest and then absorb all the food and the water out of that chyme that we usually would. Frequent diarrhea can result in dehydration and electrolyte imbalance. So looking at that process with an image, we've got our descending colon, our sigmoid colon, our rectum, and our anus. So with the mass peristalsis, we have the food moving, feces moving into our rectum. That stretches our rectal walls. This stretching stimulates an afferent pathway. So afferent arrives at the spinal cord. This is our sensory information traveling along to our spinal cord. That stimulates our motor neurons, which then go to our colon, our descending and sigmoid colon, as well as our rectum and our anus. It causes these longitudinal muscles that run down here to contract. That pulls all of this closer together, increasing the pressure in this rectum and anus. That motor neuron will also release our internal sphincter, which is made up of smooth muscle, so the one we don't control. To have defecation occur successfully, I guess you could say, we need to have conscious and voluntary control from our skeletal muscle. So we need a neuron or a nerve impulse coming down from the brain, from the spinal cord to our external sphincter to cause conscious relaxation of this sphincter for that feces to exit the body. And the last learning objective, which is a little bit of a throwback to the organ system that we covered in the last lecture, and that's to describe the hormones that regulate digestion. So there are lots of hormones that play a role in the functioning of the gastrointestinal system. Two that we have talked about before are glucagon and insulin. So glucagon, remember, acts to increase our blood glucose concentration. So we might release glucagon if it's been a while since we've eaten. If we're starting to feel a bit faint, we haven't had a lot to eat today, we need to increase the glucose in our blood. Insulin then does the opposite, so it decreases our blood glucose concentration. So say we've eaten a really big meal, perhaps it has a lot of carbohydrate or glucose. We need to bring our blood glucose levels back down, so we release our insulin to take the glucose out of the blood and store it in places like our liver. The three other hormones that we're going to talk about today are gastrin, CCK, and secretin. So if we start with gastrin, and gastrin is released from enteroendocrine cells in the gastric glands of the stomach. So gastrin comes from the stomach. It is released in response to distension of the stomach, so a really big meal, of having partially digested protein in the chyme, of having high pH in the chyme of the stomach, the presence of caffeine in the chyme, and the stimulation from the parasympathetic nervous system. Once gastrin is released into the blood, it targets gastric glands to secrete large amounts of gastric juice, to increase gastric motility, which is just the movement of the gastric contents or the food within the stomach through the stomach into the small intestine, and it increases gastric emptying. Gastrin is inhibited when the pH of the gastric juice drops below 2, and it's stimulated when it rises. This negative feedback loop is really important to maintain the optimal pH within the stomach so that we have the functioning of the pepsin, the killing of the microbes, and the denaturing of the proteins within the stomach. So gastrin, released by the stomach, stimulated by things like a really big meal, having protein and caffeine in the chyme, as well as parasympathetic stimulation. It increases secretion of gastric juice, how quickly food moves through the stomach, and how quickly it leaves the stomach to enter the duodenum. We then have cholecytokinin. You can just call it CCK. It's released by enteroendocrine cells of intestinal glands. So this one we find in the intestines, not the stomach. It is stimulated in response to chyme containing amino acids from partially digested proteins, as well as fatty acids from partially digested lipids, as well as acidic chyme coming from the stomach. Once in circulation, our CCK will stimulate the secretion of pancreatic juice that's really rich in those digestive enzymes. It causes contraction of the gallbladder, which then empties the bile in the gallbladder into the duodenum. And it slows gastric emptying by promoting the contraction of the pyloric sphincter. CCK also works with secretin, which is the next hormone we're going to talk about, and it enhances the effect of secretin. So lastly, we have secretin, and secretin is also released by the enteroendocrine cells of the small intestine. It's stimulated by acidic chyme entering the duodenum. In turn, that stimulates the secretion of pancreatic juice that's rich in bicarbonate ions, which helps buffer the acidity coming from the chyme. Secretin also inhibits the secretion of gastric juice, so we want to slow down the production of that acid in the stomach, as well as enhances the effects of CCK. So they kind of promote each other. Now that is the end for the gastrointestinal system. We've got in just under 50 minutes. As always, it's a lot to cover. All of these things, particularly these three hormones, we will go over again in the labs. I hope that you are starting to get a bit of a better understanding of the human body. The next series of lectures, we will focus on the respiratory system.